Okay. Can you see my screen?
Loading. Yes.
Great. So yes, welcome to the R42 Institute Longevity Series. This is the third series of the fourth lecture series that we've hosted. Many of you know about us already, but we are R42 group, and Ranjan Nag, Dr. Ranjan Nag is the founder of R42. He's currently also a fellow at the Stanford Center for Study and Language and Information. Teaches courses in longevity science and venture capital at the medical school. Myself, I'm a principal at R42, evaluating companies in the longevity and AI. And who are R42? So we're a fund, venture fund. Often we're first check. We invest in AI, pure AI, and longevity biotech. We also have a few other things going on, debt fund and quant fund. And currently we have around 80 portfolio companies. We're also R42 Institute, and the longevity series are part of the Institute. We host lectures like this, different workshops, also a fellowship program and founders incubation program. R42 has been inspired by the book that is called The Hitchhiker's Guide to the Galaxy. Probably a lot of you have have watched the movie or read the book as well. Here are a couple of remote opportunities at R42. So we have the Founders Program. That's our incubation program where we work with 1 to 3 companies on getting them to commercialization. And then we also have our next cohort of AI fellowship starting in June. More information on the website, but there are various AI and longevity projects for students, but also professionals all over the world. It's a very great program. We've already had 2 lectures in this series with amazing speakers. And today we have Elena Gross and Adrian Soto from KetoSwiss. And then we'll also have Dr. Marco Quarta at Rubido, talking about how to get rid of zombie cells to stay young and healthy. And then also the last lecture will be from Emil Anderson and Morten Olsted at Exceed Health, talking about male fertility. And it is my pleasure today to introduce you to Dr. Elena Gross and also Dr. Adrian Soto, who are our lecturers today. They're both from KetoSwiss. So Dr. Elena Gross is the founder and CEO. She actually had experienced chronic migraines herself, and then that's how actual KetoSwiss came about. So she would tell you more about that. And Dr. Adrian Soto is a chief medical officer as well, and specialized in the field of internal medicine as well. So, and right now, researched a lot about the potential clinical applications of exogenous ketone bodies. So over to you, Dr. Elena and Adrian. Thank you.
Thank you, Anastasia and Ranjan. It's a pleasure to be here. I will start sharing my screen. I will start off with talking a little bit about ketosis and migraine, and then Adrian will give you the good stuff about longevity and ketosis, which is probably what you've been waiting for. Can you see that screen? Is that working?
Yes, and just a little bit of logistics around the lecture. So feel free to ask questions in the chat. We will be monitoring the chat, and also feel free to chat with each other in the chat as well. And then we'll do Q&A in between, and then at the end of the lecture, and then followed by breakout rooms. So yeah, thank you.
Yes, I think we'll be maximum 30 minutes each, and then we can dive into all the questions that may arise. So I've already had an introduction. Maybe also just why I'm here is basically I used to be that person in the middle. These are my attempts to illustrate what a migraine feels like. You can't really see pain or migraine pain at all. So some of you that may not have had it don't know what it really is like. So it was quite debilitating. And I went into neuroscience to maybe within my lifespan figure out what migraines are and what you can do about them. And luckily, I stumbled across something that gave me my brain back earlier than I ever had imagined. And that is why the first patents, my PhD in the topic, and also KetoSwiss happened in the first place. So quite selfish reasons. Maybe as a quick summary, this is not about the company at all, but as KetoSwiss, just one slide, very briefly, we're basically the first in the world to identify a novel migraine subgroup. There's not really any objective subgroups before in migraine. It's just migraines, how frequent they are, and whether they're associated to your period or not. And that's or whether they have an aura phenomenon or not. That's really all the classification or subclassification we have. We identified something that we call metabolic migraine, and you'll learn a little bit more about this in a bit. And we've always also developed a preventative solution that can target the root cause of these migraines that we call MigraKete. So it's not a symptomatic pain treatment, but it actually addresses the root cause in these migraines. And we found some biomarkers, objective blood biomarkers, that can potentially predict who belongs to this group and who doesn't. And in this subgroup, we achieved 3 times greater migraine day reduction over gold standard, and that was in responders, obviously, and a small study that you will soon see. What is migraine? Well, migraine is actually the 3rd most common disease in the world. And you sitting here, you may be wondering, why the hell are we talking about migraine if we're talking about extending health span and maybe even longevity? Well, I guess that if you have something debilitating starting from your teenage years, extending a health span when you're not healthy isn't really possible. So getting healthy in the first place and preventing this from happening, that you can live long and healthy, is helpful. And it's super common. 50% of migraine patients are actually dissatisfied with current treatment options. 150 million are in the same situation as I was about 8 years ago with chronic migraines, meaning they're in pain at least 50% of the time. And it's immensely expensive. So we're looking at 100 billion direct and indirect medical costs in the EU alone. And this number is as great or greater in the US. And there's actually no preventative addressing the root cause of migraine at all. Everything we have currently in terms of treatment is just symptomatic pain treatment, meaning it's basically targeting the pain at one level or another, blocking receptors with the CGRP monoclonal antibodies, injection-based treatments that we have newly available, or just our classical painkillers, ibuprofen, aspirin, or similar. And we want to change this because these often come with a lot of side effects, and in the long run, with if you have it very, very severely, then this can even lead to things like liver failure. So you can die from the consequences of chronic migraine. Now, I want to leave you with one question, and that is, does it strike you as likely that something that is affecting 15% of the population, every seventh person in this room, does it strike you as likely that this is an accident of nature? These genotypes never had any evolutionary advantage at all. And I think we're saying that this is very unlikely to be the case. So what we're going to argue here is that migraine is in fact a civilization disease that can be prevented. Migraine is in fact, or at least used to be, adaptive. Now, in very simple terms, it's an evolutionary preserved mechanisms to warn us that there's an energy deficit or an increase in oxidative stress, too much oxidative stress in the brain happening, which would harm the organism. And the migraine then is forcing us to rest, withdraw into a dark room, and basically save energy until any energy homeostasis in the brain has been restored. And basically, in migraine, what we have is a mismatch between energy demand and energy availability. Energy demand in migraine is oftentimes greater because the migraine brain actually doesn't habituate, meaning that a healthy brain, when you, for example, look at a white wall or a stimulus that isn't changing, your brain will reduce firing to conserve energy because it's very adaptive to only basically code for change. In a migraine, that doesn't happen, so it keeps firing, which means even at rest, a migrainous brain spends on average about 20% more energy than a normal person's brain. Now we have this hyperexcitable brain, and now we put this brain and these hyperexcitable genetics into an environment today that has changed very drastically from 10,000 years ago. So in very simple terms, again, these modern diets that we're on currently leave the very energy demanding migrainous brain starving. And this is basically just what I'm trying to get here, is that migraine seems to be as much a metabolic issue as it is a neurological one. And this is actually a very good thing, because this we can fix. We can't do much about our neurological hardwiring to date, although I'm sure Ron, John, and your colleagues, you may know people out there that are already working on these things. And with gene therapy, one day this will be possible. But right now, we cannot do anything about the excitability part, but we can fix metabolism. And this is what we're trying to do. And in order to get everybody here up to speed and understand the same language, we need to understand what is ketosis. Now, you may have heard about this, especially in the US. Ketosis has become quite popular. Ketosis is actually the state where you have these so-called ketone bodies in the body elevated. Ketone bodies are small fatty acids that are produced by the liver to fuel predominantly the brain, but also the heart, when glucose is not very available. Now, the thing is, we cannot really store glucose very well in the body. So whenever we're fasting, or we used to be starving, or there wasn't much food around 10,000 years ago, without these ketone bodies, the human species would no longer be around, because the energy situation in the brain is a very special one. Now, for one, the brain is very energy demanding. 25% of energy is needed for the brain, whether we're asleep or awake, despite the brain only making up 2% of its mass. Second, the brain, despite needing so much energy, cannot actually store energy. So it is dependent on the rest of the body for its energy supply. Third, the brain is protected by something called the blood-brain barrier, which means now only 3 very small molecules can make it into the brain to fuel it adequately. There's not many things like other organs, muscles can use fatty acids, protein. The brain can't really do this very well. So we're left with 3 molecules. One is lactate. Lactate is only produced in larger quantities when we do exercise. The second molecule is glucose. Now, glucose we cannot store very well, only for like 24 hours, and then those reserves are used up. So we are in the need of a third molecule. And that molecule or class of molecules is called ketone bodies. And it's very smart. The body can make it out of our fat stores. So even the leanest of humans has 6 weeks supply of this sort of molecule to fuel the brain and the heart when fuel isn't really available. Now, ketosis is the state when these molecules are elevated. And maybe as a fun fact to remember, humans are born in ketosis. So babies still make ketone bodies. Mother's milk contains a precursor of ketone bodies because ketones are essential for brain development even after we're weaned. And it's only when we're basically put on modern Western diets that our bodies forget how to make these molecules and are not producing them in large quantities anymore. Okay, so ketosis seems to be a good thing. How do we get there? I already touched upon the first very simple way to get into ketosis. That's basically fasting. Now, fasting is endogenous ketosis, endogenous meaning the liver, the body is making those ketones with calorie restriction, quite severe calorie restriction in that case, if you're not eating anything. So this cannot be done inadequately, right? This can only be done for a short period of time. This is why about 150 years ago, something called the ketogenic diet was developed, which is basically endogenous ketosis without calorie restriction. So you can, if you do it very well, in theory, do it forever. And this basically means that you're mimicking fat or fasting in a diet where you consume about 75 to 80% of your calories from fat, moderate protein, 15% or so, and then 5% from carbs in the traditional ketogenic diet sense. So your body basically doesn't know, does the fat come from in or outside, and it switches on the same or similar pathways and metabolism. So you can get into ketosis this way, but this is a little harder. As you see here on the plate, you have to watch what you're doing. You don't get a lot of fruit. You don't get to eat rice, pasta, potatoes, starches, many colorful vegetables, and you have to watch protein a little bit as well to stay in this state of ketosis. Now, in recent years, there has been a third possibility how to get into ketosis, and that is the possibility of ketone body supplements. Now, you may have heard of these exogenous ketones before as well. There's several different ones. The first way to do this was coconut oil. Coconut oil contains small fatty acids called MCT, middle chain triglycerides. They're mildly ketotic. Now there's MCT oils that you can put into your salad or your coffee. And there's other substances called ketone esters or instant ketosis. Say the simplest way to put your body into a state of ketosis very quickly is that of so-called ketone body salts. Now, beta-hydroxybutyrate, the molecule you see here, is the most prominent ketone in the body at a ratio of something like 4:1, 3:1, all the way up to 6:1, depending on person and state. And you can bind that beta-hydroxybutyrate molecule to any kind of a cation. This could be sodium, potassium, magnesium, or calcium. You mix that, and then you can drink that as a powder and get into a state of ketosis without actually changing your diet. Well, I'm not going to promote that you are on a pizza, pasta, and ice cream diet and you just supplement with these ketones, but you can definitely be less strict when you do so. And this is what we did about 4 years ago in our first study. I think that was the first one, clinical trial, looking at ketone bodies in migraine prevention. And what we see here, migraine, I already touched upon this, is a very multigenic disease, very heterogeneous. Many different pathways together are influencing migraine phenotype, so the migraine disease. So you have a lot of small polymorphisms in genes coming together to lead to migraine. And I think this is true for a lot of neurological diseases that are common. It's not that one mutation, that one rare mutation, that accident of nature that you see, for example, in muscle dystrophies or really rare diseases that lead to a disease. It's many different things adding, multiplying together, leading to a migraine. So my migraine might be very different to somebody else's migraines, but these are 8 key pathways that have been identified to play a role in migraine, which is the microbiome and digestive health. Inflammation plays a key role in migraine. We already touched upon ATP levels, so this energy currency in the brain being reduced, as well as glucose. Excitability is increased. We touched upon this briefly. Mitochondrial functioning is decreased. That's the powerhouses of the cells. Glucose transport here, GLUT1 transporter can be decreased. And very importantly, also an increase in oxidative stress. And the good thing about the molecule that we just talked about, which is DBHB, has been shown to potentially be able to positively influence every single one of these mechanisms. So it's not the standard classical biotech approach that we're using here, where typically if you want to treat a disease in pharma, they will design a small molecule that binds to one single target only, an artificial molecule that is designed in a way that it can be patented, of course, and also only influences as little as one target pathway enzyme even. And then we're trying to solve complex civilization diseases that are caused by so many different polymorphisms in genes, and that they together lead to the phenotype. I think that will probably never work. So the beauty of using an endogenous metabolite is that it can actually influence so many different pathways at the same time, which may then have a much greater efficacy across different patients with different subtypes or slightly different genetics coming together, leading to a similar phenotype. Okay, so we did a trial. So I'm just saying this is a theory. Now I'm going to show you the results. We did a randomized placebo-controlled double-blind crossover efficacy and safety trial on the very first version of Migokete, which was not very potent yet. It was racemic. That just means it has half of a version that the body can't really use very well for energy. So it wasn't very potent, but the design, I think, was pretty nice. You can see here we had a run-in period for baseline comparison where we just measured migraine frequency. Then we had an active intervention and a placebo group, a washout period, a second run-in period to counteract any potential seasonal effects in migraine, and then crossed over to the other intervention. Now, the power here is that every person is their own control. And the placebo effect in migraine can be really large, typically around 25 to 30%. So it's very nice that every person is their own control. That gives you more power as well. This was in adults, medium to high frequency migraineurs, 40 of them, and the primary endpoint was reduction in migraine days. Yeah.
So sorry, Elena, just quick couple of questions coming in the chat, yes. And then basic question, migraine, and maybe I didn't quite understand what is migraine? How do you know, these headaches? What's happening in the head?
Yeah, so what is happening in the head? So the physiological correlate of migraine, we know that. So basically, it's an activation of nociceptors, which is pain sensing receptors, right, in the trigeminal pathway. Basically, the neurons that innervate the parts of the face and your skull, basically they become activated. And when they become activated, it hurts. Now, the big question is, why do they become activated? This is the golden question. And we're arguing these pain-causing pathways are being activated because mechanistically of free radicals, pro-inflammatory peptides that are released as a consequence of this energy deficient state or increased oxidative stress, which are interlinked. So it's basically a way to force us to rest until the energy homeostasis in the brain comes back. Clinically, a migraine is a headache of moderate to severe intensity, typically half-sided, but not always, and is also coming with other symptoms such as moving makes it worse, nausea, sickness, light sensitivity, noise sensitivity, smell sensitivity. Some people can get auras, which is phases of sensory difficulties or sensory disturbances, such as they go blind in half of the visual field or vision goes blurry. They can also have, this can go all the way to paralysis of parts of the body, to word finding difficult, so people can't speak depending on where in the brain this aura phenomenon is happening. So, I mean, what is a migraine and why are these pain causing this trigeminal vascular system, as it's called, why is it activated? I mean, whoever can answer this precisely and for sure probably will get quite a few science prizes. But that's our understanding. I don't know, Adrian, is there anything you want to add to that?
Yeah, perhaps just that, I mean, some people associate migraine with the idea of a very severe headache, which is not exactly accurate. Migraine has a very specific clinical definition. It's characterized by nausea, by a pulsatile pain, typically unilateral. It's typically an incapacitating pain that lasts for more than a few hours. It can last for days. And of course, it can get you bedridden for days, and hence the nature of why it is a discapacitating disease. Just to state that migraine is not just a very severe headache. Most migraine pains and most migraine patients, although, do suffer from severe headaches, but it's a very specific group of primary headaches. Primary headaches are those, in the typical classification or clinical classification for headaches, you would have primary headaches, those that are originated in the central nervous system, and those secondary to other diseases. In this case, for the two most common primary headaches are the tensional headache, which is not migraine, and migraine, which is of course migraine.
And maybe just to add it, and I think migraines are more related genetically to epilepsy than they are, for example, to tension type headache. So it's some epileptic patients will grow out of epilepsy into migraine. So that's maybe worth mentioning as well. And it's for sure. It's so like with a tension type headache, typically you drink some water, you go out, you move, right? All these things that you would do for a typical headache make a migraine worse. So it's no way to just go and sleep it off, because it's so you wake up from it when it happens, because there's no way you can sleep through that. It's too severe.
Right.
I can.
But you're I think you started off by saying the brain likes ketones rather than glucose. Does the brain prefer? How's that linked to the migraine part? Like what happens in the head? Is the energy source make a difference? Is that yes. The argument?
And we'll show you. Yeah, yes. We'll show you why as well. But so if somebody has this, several ways how this could work, right? Ketones have been shown to be a more effective energy source in the sense that they need less oxygen when they're burned, and they produce more ATP per molecule and of oxygen burned as well. So they can be said to be a bit more efficient and a bit cleaner because they produce less oxidative stress. They're also a potent antioxidant, meaning that they can catch free radicals themselves. They reduce hyperexcitability. right? And I think we can go back to this slide where all of this is basically shown here. They're anti-inflammatory, they reduce oxidative stress, they increase glucose availability, they reduce excitability, they increase mitochondrial functioning, ATP, the energy currency in the brain goes up, and they may even improve the microbiome or gut function in that sense. So, and anti-inflammatory. And I'll show you the data, our own data, where we have measured some markers for these different functions that indicates that this might actually be the case. And there's one question here: could the ketogenic diet help avoid migraines? Definitely yes. There's 3 studies now on a ketogenic diet intervention in migraine that reduces migraine days, migraine frequency during the short 1 month period of ketosis induced with a ketogenic diet by up to 75%. The problem is most of these patients do not sustain the ketogenic diet, even though it helped them so much afterwards, because they're adults and there's no mother cooking for them. And so they do eat something that kicks them out of ketosis, and that's when the migraine returns, which is why we have been looking at something that can be in addition to a less strict ketogenic diet or low carb approach, or potentially even outside of it. And I'll show you exactly. So yes, we'll come to those questions. That's a good question, Jason. I just see it. And Adrian will talk all about other benefits of those outside of migraines in a bit. But let me just show you the data that we collected in that trial. So I already mentioned the trial design. What we found that was about in the very first, not very potent product here, for those of you that know ketosis, therapeutic ketosis is set to start at around 1 millimole of BHB levels in the blood. Nutritional ketosis is set to start at around 0.5 millimole. Now, this product only just reached nutritional ketosis levels of 0.5 millimole 3 times a day. Still, we had a 30% responder rate. In these responders, we cut migraine days by 60% over placebo. This means you can add the placebo effect on top of the 60%, and placebo effect can be as large as 30%. So some are completely migraine-free, some have a bit of migraine left, but just for you to put this in perspective, everything else we have in migraine, on average, cuts migraine days about 20% over placebo. So we're 3 times better than the gold standard here. Of course, this is a small trial and needs to be replicated. I should be saying this more often. That's definitely true. That's what we're raising money for. But the other good thing is that we have biomarkers now, blood biomarkers that I'll show you in a second, which can predict who is responding and who isn't, which is helpful because there are no migraine biomarkers out there yet, or migraine treatment biomarkers. How many people became responders? 30%, so it was about 10 after dropouts. 10 are responders.
So 10% or 10 people?
10 people, 30%. 30% here.
Got it. 30%. Oh, I see what you mean. Yeah, so there it says responder rate. Yeah, got it.
Yeah. And then there was no safety concerns at all. Some biomarkers even for safety even improved. And the only side effects we had were gastrointestinal nature. So that was a bit of stomach upset, a bit of nausea, sometimes a bit of diarrhea. And then last complicated slide, and then I'm almost done, is basically the best finding for me is this one here. On the left side, you see the differences in inflammatory and metabolic markers between responders and non-responders. This slide is a bit hard to read, and I still should make it more pretty, but there's never enough time to do everything you want. Just focus on the bottom bar here. CRP is a general marker for inflammation and also a marker of general health and maybe even longevity. So CRP is a marker you want to have as low as possible. Now the responders here are 160%, have 160% more or higher CRP levels than the non-responders, which indicates there's something wrong here. Their inflammation is higher than optimal. Now on the left side here, you see CRP on the bottom. On the top here, CRP is about 60% reduced after 3 months of migraine intake. So more than half in the 3 months, despite this not being very potent. So that's definitely something that is going into the right direction. All other markers shown here, like triglycerides, fasting insulin, fasting glucose, HbA1c, long-term sugar, all of these values were decreased here in the responders, which goes into the direction of doing a low-carb diet without doing the actual low-carb diet. These are not yet significant in the small sample, I have to say that. CRP is highly significant even in the small sample, but everything is going into the right direction. So after 3 months of MigraKet intake, responders look like non-responders, which is exactly what you want. So this is kind of evidence to show disease reversal in these individuals. Not only do their migraine days go down, but the markers that predict if they're responding or not are normalizing after 3 months only. So with 3 of those biomarkers together, there's quite a high accuracy of predicting who is a responder and who isn't. Now, lastly, before I pass this on to Adrian, because these markers are not only markers that play a role in migraine, these markers we've just seen a question here, could this be a biohacking tool? I think for responders, certainly yes. Ketone bodies do a lot of things that Adrian is going into that could be a good tool for many people to ingest. Our main focus is on brain health. So I just wanted to show you what the go-to-market brand looks like. We're launching in the US in a half year or a bit less time. A brain ritual to indicate that you have to do something every day for your brain health. We're developing high-end brain health supplements or even medical foods. Now, this product will probably be a medical food, meaning that we can say migraine, we can have doctors recommend it. A medical food is something in between a supplement and a drug. It's basically let food be thy medicine, which we're very fond of, no side effects, but against or for preventing a disease. And our mission is basically to provide an effective solution to migraine and later on other neurological diseases. We're just looking at my Alzheimer's here. A question, is Alzheimer's promising? Certainly, yes. There's quite a lot of evidence that there may be a metabolic subgroup of Alzheimer's too, where brain metabolism, glucose is disturbed, and an additional or alternative fuel source may be helpful. Without the side effects of classical pharma products, based on solid scientific evidence and inspired by evolution, looking what has changed today versus 10,000 years ago. Where has our environment changed in such a way that what used to be adaptive may not be adaptive today? So making people better without making them worse. And the last slide is MigraKet, which is the go-to-market product. MigraKet contains more than just ketones. It's basically a one-stop migraine shop or nutritional powerhouse containing most micronutrients that you need to actually burn the macronutrient beta-hydroxybutyrate, because a lot of soils are deprived these days. Not everybody is eating as healthy as they should. So it's got 30 plus ingredients, all human identical, meaning all the same molecule that your enzyme in your body can already use without having to convert them, with using enzymes and energy that a patient or somebody not as healthy doesn't have. And human identical, high quality ingredients, transparent. Every ingredient is on there, natural where possible, and lab tested. And we're recommending you take 2 doses a day. Yes, so this is it from my perspective. I'm not sure if we want to go into questions around migraine immediately or whether we let Adrian talk about all the perks of ketone supplementation first. If you are interested in KetoSwiss in general, you can reach out to me now or after, and I let Ranjan or Anastasia decide on how we want to handle questions, or if you want to pass it on to.
Yeah, thanks, Elena, a lot. We'll do maybe a few minutes. Any questions? Who would like to?
Well, let's see. There's some questions. Like Miriam, do you want to unmute yourself? Maybe you can have people verbalize their questions to break it up a bit.
Yes, I just don't really understand how you can make the difference between the subcategory of metabolic migraine and other ones. How can you detect differences in those migraines?
It's a great question. So, well, for one, if you have access to blood and a lab and a medical doctor, I have just mentioned these biomarkers that you can check, and we have identified, of course, very early day biomarkers that you can check for and that can indicate whether you're a metabolic migraine or not. For example, if CRP is over 2.4, you're more likely to be a metabolic migraine. In addition, if your HbA1c is of a certain height, and so on. Now, this may be tricky to do for everyone. The other way that we're currently assessing with the University of Zurich whether we can predict who's a metabolic migraine or not is based on trigger factors. A lot of migraineurs that have had migraines for years have kept migraine diaries and basically know what kind of triggers will more likely than not lead to a migraine. For example, does exercise trigger your migraine? That used to be one of my most potent migraine triggers. Does fasting trigger your migraine? Also, again, one of my most potent triggers. Does not drinking, altitude, trigger your migraine? Then again, these are metabolic trigger factors, or trigger factors that are strongly linked with metabolism. And if you have more than one of those, then you may be more likely falling into the metabolic migraine category. Now, the third way, these are two ways of looking into this. The third way is just taking the product for 2 weeks. And if you don't feel any difference in your migraine days after 2 to 4 weeks, then you're unlikely to fall into the category as well. So these would be the 3 ways: biomarkers, trigger factors, or just trial and error. Does that answer your question?
Just one more question about that. So if you're fasting and this triggers your migraine, so how can you explain this? As when you fast, you go into ketosis?
Yeah, the thing is, most people do not enter ketosis when they're not eating for, say, when you skip lunch or whatever, when you're not eating for 10 or so hours. For most people that have not been in ketosis since birth, basically, ketosis or ketoneogenesis will not be turned on. So your liver is not yet making ketones, but you're also not ingesting glucose. So your brain is basically left with nothing. So this can then turn on a migraine in the short term. If you are regularly fasting and you can skip between fat and glucose metabolism very easily, then fasting does not trigger migraine, or does not necessarily trigger migraine. There's also some people that have problem with even on a ketogenic diet, and when they're in ketosis with fasting, because their energy metabolism is so sluggish that they cannot go without food for a longer time because something wrong with fat metabolism or generation of ketone bodies. That's more rare. But in a sense of fasting, when I say fasting here is a potent migraine trigger, it's more like under 24-hour fast, where you won't be in high levels of ketosis just yet. But it's a good question.
Questions. Do you need to have a balance between ingested carbohydrates and migra fat? Is there a balance you have to reach, or are you automatically in ketosis when you eat when you have migra fat?
Yes, with migra-keto, you are basically in instant ketosis because the ketosis comes from outside. So it puts you in a level of ketosis for about 4 hours without changing your diet. This may not be optimal, or it may be better to also reduce sugar for sure. Definitely always with migraine, reduce sugar and simple carbs, because you don't want that roller coaster of high carb, low carb, and insulin spike and so on. You don't want that, but it may be enough to add it on something like a low GI, low glycemic index diet.
Okay, thanks.
Thank you. And Jason has a hands up, so please.
Yes, thank you. That was a good answer on that last one too. So I have essentially two quick questions. So the first one would be about like side effects from taking taking BHB exogenous ketones or miga-ket. Just kind of what are you trading off? Like, you're boosting your ketones, but are you maybe giving up something else? Are you kind of trading one thing off for another a little bit? And then the second question is just kind of comparing those two products, like your product that you're developing versus, say, something like on Amazon, like a exogenous ketone salts combo powder. Like, how would you kind of compare those two would be my second question.
Yes, very good question. So the first one, in terms of side effects, it depends whether you have the racemic. The racemic products are basically whenever you produce something chemically that has different functional groups, a bit like my two hands, right? Same ingredients, same chemical formula, but because my hand has different fingers, right? No matter what I do, when I produce my hands chemically, I always get a right and a left spinning version. different orientation in space. And no matter what I do, I can never align my two hands. They're never identical in space, even though they have the same chemical formula. This happens whenever a chemical has different functional groups, like hands do. So this also leads me to the competitors. A lot of competitors, not all, but I would say 90 to 95% of whatever is available in the US now is racemic, meaning it's half of a version that is not used by your body and not produced by your body. Your body is handed, so your enzymes can only dock on one of the stereoisomers, on one of the spinning versions, not the other. The other one's then converted or used somewhere else. You know this maybe from amino acids. There's L-leucine, L-lysine. Those are the left spinning ones, and the body can only use those. The right R-lysine would not be natural. The same goes for BHB. There's DBHB and LDHB, and a lot of competitors have DL as well, and that will cause a lot of gastrointestinal upsets and taste a bit weird, and can also lead to quite a drop in blood glucose that we found, which some people may want, some people may not. Like in a migraine setting, you don't want to drop in blood glucose much, because that will exacerbate the problem. Now, with the DBHB, the human identical molecule, we don't see much side effects long term, and that was a big question. Is it, if you are on a high carb diet, and is that better or worse than a high carb diet and ketone bodies at the same time? Now, what this research seems to suggest, and bear in mind, this is only taking it for months, not years at a time, is that because it is reducing glucose, insulin, triglycerides, inflammation, it seems to be better to have glucose high and ketones high than not having ketones at all. But that was not certain for a long time, and I think it's maybe not even yet certain, but it seems like it's better to have it on top of a higher carb diet than not having any ketones at all, at least in the studies that are available right now. I don't see any side effects, but I have not seen studies that have looked at individuals taking exogenous ketones for years, right? So this study data isn't available. What we see in the short time, what I've showed you there, seems like it's rather protective than worse. And we also don't get the gastrointestinal stress anymore if you don't have the racemic. Now with the MigraKet, that's a migranized product. It has 30 other ingredients in there that are necessary for ketone body burning, and also are migraine protective and antioxidants. So it's like it's a product with lots more than just ketones in it. That's the go-to-market product. That is a difference. And it also being a medical food. We're working on, and this is not yet done, and it's very complicated, longer elevation ketone bodies that have much more substantial differences to products that are already on the market, apart from being human identical in that. But those are still in development. And they may end up being a drug. We don't know yet. Or maybe a supplement. That all depends on safety data and regulatory decisions down the line.
Okay, yeah, that makes sense. Just to touch on one quick thing you said, I mean, is the natural version of ketones that you're producing versus putting something into your body to enhance ketones, is that one going to be a little bit better? It sounds like maybe they're just different. Like if you're able to drop your carbs low, produce ketones that way versus high carb with ketones.
Yes, no. I would never, I don't think I would recommend super high artificial carbs to anyone. No supplement or drug you can ever take will offset a bad lifestyle, period. I would never advocate this. So if you're happy on a ketogenic diet, you have your ketones elevated, this works for you, perfect. There are people, hypothyroidism. A strict ketogenic diet just didn't work for me, right? It drops thyroid even lower. Some females just struggle with too low of a carb long term. If that is you and you are craving fruit and vegetables all the time, maybe you personally do better adding some more healthy carbs and using exogenous ketones once in a while. It's a very personal decision. The other thing is with migraine, you have a pretty destroyed liver a lot of times because people have been overusing medication for decades, right? With a sluggish liver, you may not even be able to make much endogenous ketones, right? So there also, it can help to get levels a little higher if you supplement a bit, or you could just use it as a tool when you're transitioning from a high carb diet to a ketogenic diet, just to not get that keto flu period, Or you can just take exogenous ketones when you know, oh, I'm going to eat out today. I don't know what's in the salad dressing, or I'm eating at a friend's and I don't want to be a pain in the ass, so I may have a spoon of rice or whatever. You can also just do it in a target approach. And know when your migraine may be more likely to occur, you can just supplement.
Thank you. Let's move on to Adrian.
Thanks for all the questions.
Thank you.
We can have Q&A at the end of the lecture as well, and then also during breakout rooms. So thanks, Elena. That was really great.
Yes. And Adrian will probably answer lots of questions that haven't even come yet.
Exactly. I think most questions perhaps fit in with some of the concepts we're about to touch, and please just confirm to me that you can see my slides. You should see KetoSwiss longevity slide deck for potential investors and partners. Okay, brilliant. Well, first of all, let me say that we have more questions and answers, and perhaps some of your questions, we share them as well. First of all, because some of your very relevant questions, to be honest and not to be pessimistic, will never be answered. We will never have a full lifespan randomized clinical control trial with a ketogenic diet against something else, because it would be unethical. It would be super expensive to do. It just won't happen. I mean, I really don't think we will see it. I mean, this would involve to randomize babies, newborns, to eat a ketogenic diet throughout their life, and then record this, of course, across, well, more than one generation. However, this has been done in mice, and this is perhaps as close as we can get to this particular answer. And what was observed, what was documented in this 2017 paper, was that the median lifespan increased by a tiny bit. But more importantly, the health span of mice following a ketogenic diet throughout their lives also improved. Now, it is something that I typically say or emphasize when I discuss pretty much any health intervention with my residents, is that I am not advocating, I am not stating that everyone should eat a ketogenic diet all the time. However, this is true for some people. And some analogy that it is, well, typically, or some mantra that it is very frequently repeated in med schools across the world, is that some interventions are for everyone, some interventions are for some people, some interventions are for no one. So a sedentary lifestyle with a high refined, abundant diet is for no one. Humans are not built, did not evolve to eat and behave like that. That's for no one. A ketogenic diet every day for the rest of your life, that's for some people, sure. For some particular clinical scenarios, perhaps epilepsy, perhaps people suffering from very severe migraines, perhaps, I am not saying that, I mean, if our physiology evolved to be adaptive, and it evolved, and this is something I would like to emphasize as well, is that please remember that we lived in a scenario where we didn't have the availability of processed foods or food in general until not so long ago. Even if our great-great-great-great-great-grandfathers killed a mammoth, it would go bad after a few days. The technology for preserving food is fairly recent. In other words, and something I typically say to my patients, our liver, our body does not know we invented the freezer, we invented the microwave, and we invented the blender. It just doesn't know, it is ignorant those things exist in our world. And at the same time, the availability of sugars was not the same like the one we have right now. Because even if we lived in a tropical region, well, fruits were a seasonal thing. We're a seasonal food. Of course, it is not physiological, and let's not touch on ecological, to have an avocado toast in Scandinavia during the winter. I mean, our body does not know that's possible. And there are strong biological reasons to believe that our nutrition changed throughout the year, particularly in non-tropical regions. And we were more exposed to beta-hydroxybutyrate, certainly more than what we are now, in a westernized diet. At least we were in and out of ketosis, at least for once or twice or for a longer period during the year. So yes, some studies in mice that perhaps will never be replicated in humans have studied the ketogenic diet throughout the whole lifespan of an animal. And what they showed was that lifespan was increased by a bit, health span was increased by a bit. This does not mean humans absolutely need to eat a ketogenic diet the whole time. Now, to move on, and the rest of the slides are based or structured according with these. I mean, this is perhaps one of the sacred texts. Yes.
Adrian, so sorry, the videos, can you put the videos off the slides? I see the videos over the slides so people can read better.
I'm not sure.
Or is it only on my screen?
I don't know, but maybe I can reshare.
No, it was on everyone's. I think you just got to move it up.
There you go. Is this any better? Yes. Oh, sorry for that.
Just so people can read the right. Thanks a lot. Sorry.
No. Thanks for pointing that out. Now, one of the sacred texts in the field or the scientific field of aging is this review in Celebrate from Carlos Lopez Otin. It's, well, you could say it's old by now, but it is the scaffold most intervention studies and reviews for different molecules and treatments for aging. It's a scaffold that everyone follows. What these guys did in this particular review was that they collected the different biological mechanisms that at the same time had experimental evidence showing that if this process was accelerated, the aging phenotype accelerated as well. And if this process was slowed down, the aging phenotype also slows down. So this is a very, very important review in the field of aging. And for example, if you read current reviews on any particular molecule, you would see that they are evaluated or considered according to their effects on one, most of the times, perhaps two of these cellular mechanisms. Now, these 9 hallmarks of aging are simultaneous, reinforce each other, and something I would like to emphasize is that we typically tend to think that all our tissues and all our cells age uniformly, that our skin is aging at the same pace as our brain, and our heart is perhaps aging at the same time or at the same velocity that our liver. And this is not exactly the case, because of course our lifestyle, our genes can play a huge influence on how fast is this aging phenotype in different tissues. So what I am going to discuss about may be more relevant for some tissues than others. And what I will show and talk about is how beta-hydroxybutyrate plays into these different mechanisms. Because the takeaway message that I would like you to remember is that of all the different molecules that are being studied right now for improving health span or improving lifespan, BHB is perhaps the molecule that acts on most, if not all, of the hallmarks of aging. You could have, for example, molecules that act on mTOR, or you could have molecules that improve your NAD and NADH couple concentration in your cells, or et cetera, et cetera, et cetera. You can have antioxidants, you can have, however, BHB is a molecule that pretty much checks all the boxes. Let's go for the first one, which involves, and you could divide these 9 mechanisms into 2 groups. The mechanisms that are involved in reducing the accumulation of damage, of cellular damage, or the mechanisms that are involved in repairing the acquired insults or injuries. And let's touch on perhaps one of the most famous anti-aging mechanisms, which is antioxidants. Beta-hydroxybutyrate is an amazing molecule. And I like to banter with endocrinologists, and I like to tease them by telling them that beta-hydroxybutyrate is the most important hormone they never learned about. And let me explain what I mean by that. The textbook, the endocrinology textbook, says that a hormone is a molecule that it's a substance, not necessarily a molecule, that's either produced or stored in one tissue and acts in another tissue. This concept is problematic because you could build the same argument for almost anything. You could say then that calcium is a hormone. You could say then that CO2 is a hormone. You could say then, and you could build the same case for many different substances. However, BHB is not only a signaling molecule, it is also a fuel. And we could enter deep philosophical debates on which one is the most important function for BHB. But what I want to remark is that it is both, and it is a fantastic form of fuel. Form of fuel defined as a molecule that can be oxidized to fuel the Krebs cycle, to fuel the metabolic needs of a cell. Not all molecules have this property. Not all molecules can be destroyed or oxidized or processed to produce or to achieve chemical energy. And at the same time, this same molecule acts as a signal to other tissues to coordinate the way different tissues deal with energy demands and energy supplies. However, and let's put an example, or let's put an analogy with other sources of fuel. Of course, sugar is a source of fuel. Fatty acids are a source of fuel, or are chemical fuels for cells. If you compare how many antioxidants are produced when you burn lactate, palmitate, versus when you burn beta-hydroxybutyrate, you produce way less reactive oxygen species when you are burning BHB. And this is because of the chemical nature of BHB. BHB has more hydrogens per every carbon. It is more reduced. And this is advantageous when the last steps for producing ATP. So to recap what we have so far, BHB is a signaling molecule that helps coordinate metabolism in the tissues. It is a form of fuel, but it is a particularly efficient and harmless form of fuel because burning anything will produce reactive oxygen species, but burning BHB produces less than other things that our bodies typically produce, such as palmitate, lactate, etc. Now, one of these other hallmarks of aging is the length of telomeres. Telomeres are the caps that are in the chromosomes. And it has been documented that as we age, the telomere length reduces. And once it's completely gone, you can start accumulating even more and more and more genetic defects every time your cells replicate. It has been documented that telomeres are, in different settings and in vitro and in some animals, that this oxidative stress plays a huge role in shortening telomeres. So if you address that with BHB, there is an indirect effect for BHB on your telomere length. Mitochondria are these, perhaps you've heard, or the classical statement is that mitochondria are the powerhouse of the cell. And as I mentioned before, BHB produces less reactive oxygen species when it's oxidized than the other things that we typically consume. One characteristic of mitochondria is that when they become stressed, they uncouple the production of ATP. And what this means is that in their membranes, they express this UCP4, which you see on the right side of the slide, that it is a protein that dissipates the electrochemical gradient in the mitochondria that helps produce ATP, which is the chemical form of energy in our cells. Well, ketone bodies reduce the expression of uncoupling proteins, also addressing or improving the efficiency of ATP production. But that's not the only thing. Also, perhaps, and this is relevant context, and perhaps you've heard something about this, hormones, classical hormones, have some form of receptor. And this can be membrane receptors. And for example, the classic example is insulin's receptor. Insulin is a hormone that binds to the insulin receptor, which is expressed on the membrane, on the cell membrane. Other hormones bind to its receptor inside cells. For example, thyroid hormones. And other hormones bind to the receptor inside the cell nuclei. Steroid hormones. Well, BHB do all three of them. There are at least 2 cloned and specific membrane receptors for BHB, which help regulate energy consumption, energy expenditure, and lipid metabolism. But they themselves can go to the nuclei and modify histones, which are the proteins that help regulate gene expression, to change gene expression. And of course, a very relevant question would be, which genes are regulated by BHB? And these genes have to do with almost anything, but particularly with chronic illnesses. So to go back to the previous point I was making, we evolved in a scenario where our genes, at least intermittently, would be exposed to the regulation of ketone bodies, in concrete of BHB. And we don't have that anymore. And there are some, well, of course, you could follow a ketogenic diet, but as Elena mentioned already, you need to keep it and to be strict about it for at least 3 days to start producing relevant concentrations of BHB, and you cannot keep it. You cannot fast forever. You could keep a ketogenic diet forever, but that's also hard in the Western world. And to summarize what I've been saying so far, BHB is a biologically relevant molecule to which we were used to be exposed more frequently, and we no longer have in our daily lives. Autophagy, which is the process which perhaps you've heard of, that's involved with the mTOR pathway. Autophagy is a process in which our cells recycle old proteins, old membrane, old membrane parts to recycle its components, lipids, proteins, and which helps clear up and unclog the glycated proteins that can accumulate and obstaculize the normal functioning of cells, is promoted by ketones. To be fair, it is not entirely clear if ketones themselves, if beta-hydroxybutyrate itself drives this mechanism directly, or if it's a consequence of fasting, because of course the most important stimuli for autophagy is fasting. And in this case, please be patient with all scientists and give us time and money to answer these questions. Because to be honest, we don't know, because it was not until very recently that we had the capacity to test these two physiological states separately. Not until so long ago, it was not possible to test ketosis without fasting or without a ketogenic diet. But another amazing thing about exogenous ketones is that they allow us scientifically to start answering which of these effects are a direct consequence of beta-hydroxybutyrate elevation and which ones of these effects are secondary to carbohydrate restriction or to fasting. So this is one of the questions we still have. Perhaps you already know about zombie cells, I heard in the beginning of the talk, or about senescent cells and about stem cells. Most of our tissues have their small populations of stem cells, which replace old and senescent cells. Well, BHB, of course, has a lot to do with those particular subsets of cells. It has been documented that prevents vascular senescence through these complicated molecules, but also that it is involved in stem cell renewal, at least in the gut. And actually, there is anecdotal evidence for ketogenic diet improving clinical outcomes for patients living with inflammatory diseases. It is not clear, because something I will address later, is that beta-hydroxybutyrate is also a powerful anti-inflammatory molecule. So it is not clear to what extent these clinical benefits are secondary to self-renewal through intestinal stem cells or to the anti-inflammatory effects of BHB. but in both cases, it is something definitely related to BHB. And as I was mentioning before, and something I also try to transmit some intuition about how this all makes sense. Try to think as Mother Nature, and try to think as we need a mechanism to warrant that all tissues will cooperate and work simultaneously to adapt to a low energy environment. It would be a very bad idea to be around or to be involved in a low energy scenario with energy restriction and to have a very expensive tissue. And BHB is this message. The only tissue in our body that does not oxidize ketone bodies is the liver. And this is because the liver produces them. So to avoid eating what it just produced, the liver suppresses these enzymes, but all the other tissues respond and have the signaling molecules that respond to changes in ketone body concentrations. So it is effectively a coordinating molecule. And to address some of the adverse effects questions, something that I would mention is that bear in mind that the normal baseline fasting concentration of BHB in our blood after an overnight fast in someone following a Western diet is 0.1 millimolar. After 3 days of fasting, this 0.1 becomes 3.0. So this is a molecule that increases its concentration 30x when we are fasting or following a ketogenic diet. Our body, it is a molecule that our body chooses to produce literally more than 30 times its baseline concentration when we are fragile in an environment. So we can say it's pretty safe. And one of the experiments in my doctorate, I was blessed by finding lots of crazy people who were willing to go where physiology has never been before. And many of the participants I had the privilege of collaborating with in my studies were people following a very strict ketogenic diet, who ran marathons fasting, who were supplementing BHB, and their BHB concentration never went above 6. Some people get confused between ketosis and ketoacidosis. They are as different as day and night. Ketosis is a normal physiological state. Ketoacidosis is a pathological state. Even if you run a marathon fasted, follow a ketogenic diet, and supplement with ketones, your ketones won't go crazy up if your kidneys work, if you keep drinking water. If you are someone living with diabetes who has been off their medication, is severely dehydrated because they are losing lots of liquid through the urine, and has super high blood glucose, this dehydration will yield you into ketoacidosis. They are very, very different. So just let's not confuse these two terms. And in a healthy working organism, your BHB will never go dangerously high. Almost to finish, ketones can improve nutrient signaling pretty much by coordinating these same mechanisms and pathways. The two most important families of or pathways they have been related to is the AMPK, which is one of the master regulators in the cell for assessing how much energy there is, and the sirtuins family, not only one, but also 3. And that's the summary, and I would be happy to take your questions and perhaps to go back to any of the slides if you want to see some of the data I was mentioning. Yeah, I'll shut up and I'll be happy to hear you.
Thanks, Adrian. I got a quick question for you. The last part you were saying, I was a bit confused. You were talking about like ketone levels, and then you said, you know, you're able to increase them 30x, and then you were talking about BHP levels. It sounded like not going above 6 to then not going to ketoacidosis. are those two different measurements, BHP levels versus?
No, sorry, I misspoke. It's BHB, beta-hydroxybutyrate.
Okay.
Beta-hydroxybutyrate, yeah.
Okay, so going from 0.1 to 3, that was BHP.
That was BHB, beta-hydroxybutyrate.
Okay.
Beta-hydroxybutyrate baseline concentration, 0.1. Beta-hydroxybutyrate fasting concentration after 72 hours, 3.0.
Okay.
In George Cahill's classic experiment, it was more like a jackass anecdote than a proper experiment. His fasting monks, after 20 days, had precisely 6.0 of beta-hydroxybutyrate.
Okay, and that 0.1 was after a 12-hour fast or like right after dinner?
That's correct. No. 12-hour fast.
12-hour fast, okay.
Our physiology is somehow different to other animals. For example, octopi have ketone metabolism. Rodents, of course, have ketone metabolism. We take slightly longer to get into ketosis. Rodents typically enter ketosis after a 12-hour fast. But because we have good reserves of glycogen in our liver, we take longer than that.
I see. And is there like 0.1 is obviously pretty low, but like, what would you say you start kind of reaping like some of the major benefits from an aging perspective, like a level 1, maybe level 2, level 3?
This is an open question. Sure. I mean, this is a very relevant, and to be honest, I don't have the answer I would like to give you, because no one has done proper experiments to pinpoint the concentration where these things start to happen. However, the closest I can get to that answer is that it has been documented that after 0.5, our brain starts using ketone bodies, and it starts. Let's also mention that not all tissues use beta-hydroxybutyrate in the same rate. The 3 tissues that use most of BHB are the brain, the kidneys, and the heart. All other tissues can burn ketone bodies, but not at the same rate. And we could say that most tissues will spare glucose for the brain, and if needed, beta-hydroxybutyrate for the brain. Because, of course, we evolved to privilege our brain.
So if you're at like that, say you're at that 0.5 level, maybe you took some supplement, some BHB supplement, but you still have glucose. Would your brain and heart and kidneys choose the glucose in that case, since you still have some, you haven't burned that off? Or would they?
This is this is a fantastic question. Yes and no, as most things in biology, I guess. Bear in mind that in contrast with glucose, ketone bodies do not need a specific transporter into the cell. Glucose in the brain has GLUT1s, which are constitutive, and GLUT4s, which are expressed as a signal. So when you are eating a carbohydrate diet, your neurons and your astrocytes express GLUT4, and more glucose can enter. Some of the things that has been documented in most neurodegenerative diseases is that GLUT4 and mostly GLUT1, the expression of these 2 glucose transporters is reduced. So on top of being ill, those neurons are hungry. The advantage of ketone bodies is that they do not need an intermediate molecule to induce the expression of their specific transporters. They can use the monocarboxylate transporter family. They can use at least 4 of those transporters to get into neurons. In other words, if you have both ketone bodies and glucose at the same time, your brain will use both. This is not true for working skeletal muscle. Work from David Dearlove, who was a colleague of mine in the same lab I did my doctorate. He precisely answered this question 4 years ago. If you give to the working skeletal muscle at the same time glucose and beta-hydroxybutyrate, it will use beta-hydroxybutyrate up to its 25 maximal working capacity. After that, it's all about glucose. let's remember, and something I typically say when something I typically say when I am teaching exercise physiology or biochemistry is that running is not the fast version of walking, metabolically speaking, at least. Because running, a sprint, is pure carbohydrate. It's pure glycogen. It does not matter how much fat there is in your blood. It does not matter how much ketone bodies is in your blood. If you are pushing your muscle, your skeletal muscle, to the near maximum, all it will use is glucose, regardless of what's around. If you are working at a lower pace and have ketone bodies, your skeletal muscle will most likely use fatty acids and ketones. Another good example, and perhaps, I mean, of course, this was not properly measured, but I think it's indirect evidence of this fact. For example, if you compare the physiognomy of Usain Bolt, who is a very muscular guy, who runs very fast, versus the physiognomy of Eliud Kipchoge, who is a very lean guy who also runs very fast, they're very different. One is more muscular than the other. And if you compare, for example, I don't know if you're familiar with Zach Bitter, the ultramarathonist, he was only able to break his own world record when he engaged in a ketogenic diet. But because he's running at a very slow pace. If he tried to sprint, I am sure, I mean, most people could outrun him, but because they are different intensities use different fuels.
I see.
I don't know if I answered your question or
no, I think you did. I mean, the thing that surprised me was, I mean, I guess obviously if you don't have any sugar left, if you're sprinting, you would then have to burn ketones if you were fasting, right? Like if you're at a marathon while you're fasting.
Let me add one thing here quickly. That's worth mentioning. There's always going to be glucose. If there is no glucose,
yes,
you're
exactly.
dead, right?
Exactly.
So it's the normal state that there will always be ketones and glucose at the same time. You will never be at a state that there's no glucose or you're dead. Even the brain requires like 30% at least of its fuel demands from glucose. So you're always burning glucose and ketones at the same time. Even in the tissues that may prefer ketones over glucose will always be there. The question is in what kind of a range and what is taken up first. And Adrian has done a good job in explaining that, but it's good to know that there's always going to be glucose and ketones at the same time.
And for what tissue? Because, for example, glucose is never zero. And if you are someone struggling with a neurodegenerative disease, you have perhaps a normal range of glucose, but your brain does not have the right transporters for that glucose. So it is energy deprived. And if on top of that, you are never into ketosis, well, your brain is basically starving. So that's when it becomes more evident. The fuel part or the fuel function of ketone bodies, but let's not forget, it's not their only function. They're very powerful, relevant, and widespread signaling molecules.
How about that third source of fuel? I'm not as familiar. You say like lactate when you exercise. Like, is that a good source for the brain, or is that just kind of a third one that's not used that much? Or
the brain, something that's particularly true for the brain is the lactate shuttle. The brain is arguably our most precious tissue. And consequently, its nutrition is quite specific, is definitely more inflexible than other tissues. Like, I mean, your muscle can burn anything. Your skin can go through a lot of things, and the brain can not. So neurons can and consume lactate, but once it's pre-processed by astrocytes. So also, let's remember that when we are exercising, the fuel priorities change mostly to your brain and your skeletal muscles. So, for example, blood leaves your gut, blood leaves your skin, or I mean, it never goes fully away, but the flux is shifted to these more relevant tissues. So lactate can fuel the brain, yes. As directly as ketone bodies, no. This does not mean that its consumption is zero by the brain, but it does mean that ketones will be privileged in this case because your lactate will more likely be shifted to your other lactate-needing muscles, such as skeletal muscle. This is particularly true when you are working out.
Gotcha. No, makes sense. And can you actually run a marathon while fasting, or is that?
Oh yeah, for sure. Yeah. I mean, I have a friend of mine, he's an anesthesiologist, and he runs 100K every 3 or 4 months, absolutely fasted. After a fast of 36 hours, he has done this multiple times. I've had a blast poking his veins while he's doing so. And yeah, I mean, this has been, and Zach Peter ran, I think his record is something crazy, like 100 miles in a super short time frame. I mean, I don't want to make a mistake and say something wrong, but my point is that if you compare the records for the 100K mark, which he held as well, the difference when he started a ketogenic diet is, I mean, it's something like almost an hour or something. Like it is a huge difference. But because he's running slowly, when Sean, my friend who does this, jumps on a bike on a ramp test, he does not perform as well as people who are carb adapted because, well, he's been living with a ketogenic diet for a while now. So my point here is our physiology is adaptive. However, we were definitely more exposed to ketone bodies than what we are now. And this is something that perhaps we should bear in mind if we try to. Some people say or ask me if I am afraid of dying or why I am so obsessed with aging. And I typically say that no, I mean, the point here is to die young as late as possible, to preserve your health span for as much as you can. And I think we need to acknowledge that despite our amazing technological progress, we still have the metabolism of a caveman.
Yeah.
Yeah, thanks, Adrian. Let's give opportunity to others to unmute themselves. Jason, thanks.
Thank you.
Anyone else? There are some questions in the chat as well. Maybe Miriam, Henry.
Sure, I'll go. Just yeah, boy, so much to ask. I'm a journalist, so of course I have billions of questions. 400-meter dash is apparently the longest sprint in sports competition. That's what, a quarter mile, 400 yards, 4 football fields long. Is there anything we can say about studies with regard to these athletes and their diets and their brain function? After all, we started with talking about migraines and pain and the such.
I mean, absolutely, and this is a fantastic comparison because that's spot on. If you plot the world records for the 100-meter sprint versus the 200-meter sprint versus the 400-meter sprint, they are typically done by the same athletes. It's not rare to find that the same athlete is competing in all 3 trials, 100, 200, and 400. But when the same athlete tries to go a bit further, the 3K, the 5K, they just, I mean, they not even qualify for those. And the athletes that perhaps top the podiums for those track distances are not as good for sprints. This has something to do with blood acidity while we run. And this was perhaps best studied by UCLA 10 years ago, where they measured the blood acidity of pre-Olympians right before the sprint and right after the 100-meter mark. To put this in context, let's remember that blood's acidity is measured in the pH scale, which is logarithmic. So each unitary change is actually a tenfold change. I start getting worried when my patient's pH is below 7.3. They measured 7.4, which is a normal pH, before the sprint, and 10 seconds after, they observed 6.9, which is, of course, incompatible with life if sustained for longer. The rate-limiting factor for the 400 mark seems to be blood acidity. Our tissues basically start choking. If you try to sustain the same intensity and the same acid production for a sprint longer than that. Every time someone has claimed that this mark will never be broken, an athlete shuts our mouth, typically within less than a decade. This was true, for example, for the 2-hour mark and then you keep chugging. And I mean, I don't want to say that it is impossible to run 400 meters in less than 36 seconds. Perhaps someone will achieve this one day, but I am highly skeptical that you could keep the same pace you have for the first 100 meters and for the last 100 meters in the 400 competition because of what has been observed with blood acidity. What we also know is that, I mean, of course, it would be impossible to make these guys run at that pace with an EEG plugged to their foreheads. We would love to do so, but again, it will never happen. What we can guess is that the only two functioning tissues for those 10 seconds are their brains and their muscles. We also know that they tend to be, well, most extreme or top tier athletes are somehow metabolically inflexible because, of course, they try to, even if they don't know it, they try to privilege the metabolism that's best suited for their particular sport. Not all sports use the same fuels, and nutrition should definitely adapt. It's not at all the same to cycle in a closed track, which was, for example, one of the world records beaten with exogenous ketone supplementation, than to mountain cycling. Despite there's of course an analogy in both scenarios. So to shut up and move to another question, I just want to summarize saying that exercise can have very different metabolism pathways involved, even if it looks fairly the same.
Thank you. Thank you.
Adrian, there's been one question on Alzheimer's and all this, so maybe I can just quickly show one slide, if that's okay, on this question or cognition at least. And then we can move to I know that Pam is patiently raising her hand, so we'll go to her after. And this was now I don't see the chat anymore, but basically the question was, are there neuroimaging studies that show changes in cognition with ketone bodies? There were really early studies here where you can see on the bottom, you basically see mild cognitive impairment, which is an early stage of Alzheimer's disease or dementia. You can see when they're given anything that is not ketotic. So I think in this case, it was glucose or a longer chain fatty acid. You see the brain in the scanner here is pretty silent, not much is happening. But when you give as a comparison, you give ketone bodies here, in this case, middle chain triglycerides, which are very mildly ketogenic, the brain lights up again. So this brain that cannot use glucose very efficiently can actually use and burn ketones. So you can see they can get into the brain and they are being burned where glucose is not. And the interesting bit up here is these are serum levels of BHB. So the higher the BHB levels from this middle chain triglycerides, the better the change in recall. So the better the cognitive improvement in that early study. The first one was Riger et al. And then Franchi et al. In 2019 kind of replicated these findings on cognition. And I'm going to stop share, and Pam, floor is yours.
Thank you for this. It's been really fascinating to see the influence of BHB, not only in metabolic migraine, which I believe one of my friends might qualify as suffering, but also in the aging space with you, Adrian. And I'm just wondering, given how BHB's impact on the 9 hallmarks of aging and also on the migraine situation, is there a protocol that either one or both of you have considered would be the correct supplementation if you were taking this from an aging perspective? Health span perspective? Any ideas?
I don't know if Elena wants to go first, or
I can
go first.
I can say a few things that immediately come to mind, and then Adrian probably can complement or answer this properly. I have developed such a protocol, or a variety of protocols for migraine. It's very individual. That's the first thing. So I'm not even sure at this stage that everybody profits from the presence of ketone bodies. We've seen that in the migraine group, I think around 50% profit. For the rest of them, it really looks like it doesn't matter whether they're taking it or not. I assume the same will be in the non-migraineurs. There's probably, maybe it's 50%, maybe it's a different percentage of people that will profit from the presence of ketones, and the others will not, in the sense, or not as much, right? It's always a continuum of some profiting a lot and others may not really benefit at all because they're at a ceiling when it comes to metabolism, a ceiling effect, potentially. So there may be a few of us that don't even benefit much at all. We don't know. I'm just hypothesizing because we don't know whether 30 years down the line somebody may not have gotten Alzheimer's because they've supplemented 30 years earlier. We just don't have that data. And as Adrian says so nicely, I don't think we can really gather that data because it's not ethical, and it's also not really affordable to do studies for decades. I think, so for migraine, I have a 4-step protocol. In order to be able to even burn ketones, you need to have all the micronutrients present. So I always say, check your blood levels, check that you have all the minerals, all the vitamins, every coenzyme, everything you need to actually have a well-functioning mitochondria is working. Second, I think everybody wants to have fairly stable blood glucose. I think that's, again, something that's probably going to be true for everyone. Some people, for that means they can still eat pasta and potato, and their glucose metabolism is so good that they're kind of okay. Others need to watch out. So the best way to find out might be wearing a glucometer for 2 weeks and see what foods are really spiking your blood glucose. So keeping that stable. The other thing is adding, especially if you're vulnerable to migraine or want to have a long life, having more antioxidant capacity than oxidants is key for keeping mitochondria healthy. So this is whole foods, no processed foods, getting rid of stressors psychologically, chemically, so maybe ideally organic, getting rid of all these crappy cleaning products. Don't put on your skin necessarily what you wouldn't put in your mouth. That's my rule for migraine. Others may have other flexibility. So this is antioxidant. That's the third pillar. The fourth pillar is adding this more efficient fuel source if you need it. This could come either with a ketogenic diet or cyclic ketosis. This all depends. There's not one protocol for everyone, in my opinion. So it could be cyclical ketosis. Some people do it because I think their ancestors come from Europe or whatever. They say, well, in the winter months, I'm more in ketosis because that's what naturally would have happened 10,000 years ago. Others say, well, I'll do ketosis for a bit once a month. Others say, well, I do a week every 2 months or something. This can be very individual. I think being in ketosis once in a while is a good thing. You can do this with the diet and or supplementing ketones, and then again, individual. You could say, I'm traveling, now I'm very vulnerable, I'm at a party, I want to eat carbs and maybe sugar, so I'll supplement ketones today. Or you say, I want to, my mother has Alzheimer's and I think I'm vulnerable, I'm going to take this every day because I don't want to take the risk. So I don't think it's just, something for everyone, but I think these 4 pillars should be watched for longevity. Having all the micronutrients you need for energy metabolism, getting blood sugar stable, having enough antioxidant capacity, getting rid of all these stressors, and then getting an alternative energy substrate for your brain, at least occasionally, endogenously or exogenously. And now, Adrian, sorry, long-winded answer.
No.
Thank you, though.
No. Perhaps the only thing I would add to that is to emphasize that the non-eating hours are as important as the eating hours. And I think that we definitely did not evolve to have 3 meals per day with a super large carbohydrate-rich dinner. There are multiple independent pieces of evidence that suggest that intermittent fasting is perhaps, or some form of time-restricted feeding is perhaps the best way our body can handle fuels in general, regardless on if you are super keto strict or just, some people like to say they eat low and slow carb. meaning they eat less than 150 carbs, and all of those carbs are low glycemic index, low absorption, which is also very, very feasible. Regardless of what you eat, I think it's reasonable to state that, yeah, it shouldn't be 3 times a day with refined carbohydrates in all of them. Cake is only for your birthday, and perhaps full stop. You know, like I'm not saying never have cake, but well, only your birthday. The problem now is that Facebook will remind you everyone's birthday every day, so avoid those daily excuses for cake. And that's it. That's it. I also believe that we should be mindful that our diets and dietary patterns should match and coordinate with our daily routine and our job and our schedule. I mean, this is also a very important part of diet. And for example, I've been fasting every Monday for the last 4 years. I don't eat on Mondays, almost never. And this is just because Monday is my busiest day of the week. And it is actually helpful to have a few extra hours those particular days. And it's also, of course, a form of time-restricted feeding. I typically eat once or twice per day maximum. It's typically OMAD, only 1 meal a day. And all I am saying is this works for me. What I try to achieve with my patients is to understand their routine, their likes, their dislikes, and to match something that works for them. In the end, of course, we need to practice evidence-based medicine, but it is not rare to find patients who do not match the inclusion and exclusion criteria of those studies. So in the end, if we want to practice personalized medicine, which is preferable, we need to adapt to N=1 trials and to find what works for the person in front of you, and not necessarily what works for everybody all the time, because arguably such thing does not exist. There's nothing that works all the time for everyone, for every disease.
Thank you, Adrian. On that note, we will go into breakout rooms, and some of you might be in the same breakout room as Adrian and maybe Elena, although Elena might have to leave sooner. So we'll go for maybe 15 minutes, but feel free to leave earlier if you prefer to. Thanks, everyone. Okay, 1 second. Great.